Welcome back, everyone, to this third webinar in the ARC Clinical Incubator Program series. And in this webinar, we will be discussing research methods and how to draft the method section of your manuscript. And what I want to cover in this webinar is why the method section is foundational to writing an effective clinical or epidemiological research paper. I then want to touch on some common elements and structure to consider when drafting the methods section of your upcoming manuscript. And then I want to highlight how we can actually use the Strobe guidelines and checklist to ensure we are writing a strong methods section and we are including all the information that's going to be of relevance and importance to our readers. So people's descriptions of the purpose of a method section certainly may vary, but in general, most will agree that the overarching goal is to provide a transparent description of how a study was conducted with sufficient detail to allow others to understand and to reproduce your work. Other purposes of the method section can include allowing others to compare their research findings to yours. So for example, to be able to put your research into the larger context of information known about a given topic. So for instance, when they are doing their work and they want to cite your paper and describe it in the discussion, there should be sufficient information there for them to be able to do this. Other reasons might be to combine your results with similar studies. For example, if someone's carrying out a meta-analysis and they want to include your paper in their study, there should be sufficient information in your methods section to allow them to extract the pertinent information to be able to do that. And then in some cases, some of your readers may actually want to apply your methods or results in practice, right? So they're looking for the translational relevance of that work. And so essentially, the method section enables others to critically evaluate the validity of a study's approach and their results. And it also gives us an opportunity, an important opportunity, to highlight that the study adhered to ethical principles and guidelines. So this should be a statement that we always include in our methods section. So I say the method section is foundational because it really is your only chance to describe the who, what, when, and where of a research study. So this is really your only opportunity to explain to your readers how you generated your findings and how you ultimately came to your conclusions. As I said before, it allows readers to evaluate the validity and rigor of a study, to judge the credibility of those findings, and it gives others the information to attempt to reproduce your work, right? So this is a hallmark of the scientific method and critical to scientific inquiry in general, right? That level of detail is an expectation when conducting clinical or epidemiologic research. And really it does give you the opportunity to showcase your expertise and command over the subject matter in which you are working. Well, when it comes to common components of the method section, I sometimes joke when you've seen one method section, you have seen one method section, but certainly there are generalities across papers and in terms of how you may want to structure your method section to provide the flow of information in a way that your reader is going to be expecting to see that. And so my first piece of advice is that components will often differ, I should say, across academic disciplines and journals. So it is always of benefit to actually check the author guidelines of the journal to which you think you may want to submit your manuscript. Often they'll have very detailed information about some of the sections of the manuscript and the format they want things to be in. And so often for some journals, they will have detailed information about what they want to see in the method section and even the subheadings and subsections for which they want you to break that information out. When that information isn't available, another piece of advice that I have is certainly to look for several papers from this target journal of interest and to look at those method sections, to read them over, and to see how other authors are outlining their method section, and to use that as a template for when you're conducting the writing of your own paper. That being said, for clinical research studies, we do often see a few kind of standard subsections in which the method section is broken down into. And these can include subsections including the study design. You'll have a section often on the study population and the setting. For randomized trials, which I don't believe will involve any of our papers, but you will often have a section on the randomization and how the intervention was carried out. For observational studies, you will certainly have a section on your data sources. This will be information about how your data were obtained, who they were obtained from, perhaps what instrumentation was used or tools to collect that information. And then you will certainly want to be able to describe your primary and secondary outcomes to your readers, as well as having a section describing your primary exposures and measures that were of interest in your study. And then typically most method sections will close with a sample size determination and a statistical analysis, but those will be the topics that we cover in the next webinar. And so now I want to touch on a few of these sections in a little more detail to try to give you a feel for some of the information that you might want to include in each of these sections of your methods, and also to provide, I think, a few good examples, maybe to help get you started, and maybe in some cases help you understand where you can actually access some of this information when you're working on large studies, such as the ARC project, or if you've already drafted the protocol of your study. Often you've already written much of this information. So when it comes to the study design, I think this is often a fairly straightforward section of the methods, where we clearly want to state the type of study that we carried out, right? So this could be described, this could be a randomized controlled trial, if we were to carry out a trial, or a cohort study, which I think most of you will probably be conducting some analysis of this existing cohort. Or perhaps a case control study is an apt description, depending on how you selected your cases and controls. And so I think a good example of this is actually found in the ARC protocol. So if you have the protocol, you can look at section 4.1, where our investigators clearly described, I think, the study design very well. And so an example here is that this is a prospective international cohort study designed to recruit ARF cases and for age-sex matched control groups to further our understanding of acute rheumatic fever risk, presentations, and outcomes. So I think this is a good sentence found in the protocol that could be adapted very well to fit the description of the study design for a given paper. When describing the study population and setting, you want to be sure to describe where the study was carried out. For instance, was the study carried out in a hospital, in a clinic, or in the local community? And part of that could be, where were your patients or your participants recruited from, right? That should often, almost always, be included in your description of the setting. We certainly want to specify the inclusion and exclusion criteria for our participants, right? So what were the eligibility criteria for someone to be able to participate in our study? And then, what were also those criteria for which we would have to unfortunately exclude a participant from being able to participate? And then, as I mentioned before, we want to describe exactly how participants were recruited and how they were enrolled into our study. And again, I think you'll find much of this information in the ARC protocol. Here at the bottom, I'm showing you an example of sections 4.2 and 4.3, where you can see some of the study setting information for the ARC Diagnostic Core Network. And you can see here some of the physical locations, for example, for which participants were recruited. Many of you, I'm sure, are familiar with the inclusion and exclusion criteria and drafting these for research studies. But a good description is also found here, for instance, of possible acute rheumatic fever. And so the inclusion criteria could be ages 3 to 18 years of age, and also presenting with at least one of the following: joint pain of unknown etiology, plus or minus fever, suspicion of rheumatic carditis, suspicion of chorea, or known RHD with suspicion of acute rheumatic fever recurrence. And so these would be, again, examples from the protocol that could be adapted to help you describe or to start writing your method section. When describing our study outcomes, my general advice is to start with clearly defining the primary and secondary outcome measures of your research study, and then to describe how and when those outcomes were assessed. Right? So time is often a critical component of a clinical or epidemiologic research study. And so we want to make sure that this aspect of time is clear to our readers when writing about how we assessed our outcome measures. And then we want to specify who actually performed the outcome assessments. Was it the clinicians in the study? Did the patients self-report their outcomes? Did members of the study staff actually objectively measure these outcomes? Right. So to give a little detail of how this was actually carried out in practice. So here at the bottom, I've got what I think is a very well-written example from Dr. Beaton and many of our colleagues in the ART network, where they describe an outcome as the primary outcome was echocardiographic progression of latent rheumatic heart disease at 2 years. All participants underwent standard echocardiography at enrollment and at age 2 years. We assembled a 4-member consensus panel to evaluate and classify echocardiograms on the basis of the 2012 World Heart Federation criteria. Right, so I think this is a great example because they are clearly hitting all 3 bullets that I described above and providing a very well-written depiction of this primary outcome that will be very clear to readers of their paper. So when it comes to describing our exposures or our covariates that will be used in our analysis, I think this follows very closely to how we describe our primary outcomes. So I like to start with clearly defining my primary exposure or exposures for a given analysis, and then also any covariates that are going to be included in our statistical analysis as well. And so to do this, I think we want to give our readers sufficient understanding of where those data came from. So what were our sources of data, and what were the details of the methods of assessment, right? So how do we actually go about collecting that information? And so we want to describe this to our readers in detail. And we want to highlight if and how instruments may have been used in our study were validated in specific populations, right? So if we are using a validated instrument, we certainly want to mention that, and we want to be able to cite that instrument, right? So to cite the paper that did the validation for that instrument. And that certainly is to give credit to those investigators who developed that work, but also to clearly point our readers to more information on that instrumentation. In instances where we are developing a new method or a new method of assessment, then I think we certainly want to devote more of our method section to the description of that method and to that approach, right? So that's generally my approach or thinking on the topic, is that if I'm using information from a well-defined data source or using a well-defined instrument to collect this information, it probably requires a little bit less information and space in the method section. But if I'm doing something novel or for the first time, then I really want to provide a detailed description of how we went about measuring and using that information. And certainly if there were any approaches that helped us reduce bias or measurement error, then we want to include that in our methods. And here at the bottom, I'm showing you an example. So this is a fictitious example that I just wrote here very quickly about maybe how we would describe the exposure of smoking in an epidemiologic research study. And so here I write, smoking exposure was assessed at baseline using a standardized self-administered questionnaire. I could actually cite this if this questionnaire had been used before. And participants reported their current smoking status of either never, former, or current smoker, age at smoking initiation, number of cigarettes smoked per day, and years of smoking. For current smokers, the type of cigarettes, whether filtered, unfiltered, methanol, etc., was also collected. Former smokers provided information on their age at quitting and cumulative pack years of smoking history. To validate self-reported smoking status, a random subsample of 100 participants underwent biochemical verification using saliva-derived cotinine measurements at baseline. Right, and so my main point here is that as opposed to just saying we measured smoking status, to write a good method section, a strong method section, we want to make sure we give the type of detail and information that I'm giving here. And so some general advice when writing your method section that I think could be of help is to always think about your target audience, right? Who are you writing to? What do you want to convey? And then write directly to them, right? Often when I'm writing my method section, it may depend on what kind of paper I'm writing. Is my audience more, am I envision a clinical audience or more of a research audience, right? Often it's a balance of those. But sometimes you'll have some idea, right, of who you may envision is reading your paper. And when you hold that person in or persons in your mind, often it's very helpful to think about the level of detail and what we want to emphasize. Again, always with this idea in mind of giving them enough information to reproduce my work if and when they wanted to do so, right? So to me, that's kind of the standard that I'm always trying to hold myself to. I think it's very helpful to always pull information directly from the study protocol when at all possible, right? Often you've already thought about this, often you've already written much of this information down, and so very often you can just go back to the study protocol and use that information as a starting point when writing your method section. As I said before, always be sure to cite previously developed methods and approaches. And certainly when you are working with a newly developed method, as I said before, to provide sufficient detail. And sometimes now this may come in the form of an appendix or a supplement to your manuscript. Since we have already done the research and we are now describing it to our audience, the method section should always be written in the past tense. And then this last point is one that I see broken sometimes, but we certainly don't want to try to sneak anything into the results that is first not described in the methods. Often I'll see this in terms of new statistical analysis or the result of some analysis that wasn't described well in the methods. But this could be certainly including a measure or some exposure or some outcome that we didn't first describe in our methods, right? Including it in the results. And so anything that we see in our results, or certainly anything that we discuss in the discussion of our manuscript, we always want to make sure we have first introduced it and described it in sufficient detail to our readers in the method section. And the last point I want to make here, or at least the last piece of advice that I want to try to convey, is that when I'm writing the method section of an epidemiologic research paper, I always make sure to have a copy of the Strobe checklist next to me. And as I go down the method section, I verify and make sure that I've hit all the points outlined by the Strobe recommendations to ensure that I'm writing a strong method section and that all the information that's important to convey is being included. And so for those of you who aren't yet familiar with the Strobe guidelines, This is the Strengthening the Reporting of Observational Studies in Epidemiology Guidelines, which are a set of recommendations for the reporting of observational studies, right? So these were developed by a panel of experts with extensive experience in this space, really with the purpose of providing guidance on how to report observational research well, and to ensure we are transparently and completely reporting the information that's needed to convey to our readership for an epidemiologic study. Right? And so with these guidelines, which are, I'll provide a link to in the additional resources, they also provide a checklist. This is very, I guess, pragmatic, very functional. This is just a document that includes a list of items that should be included when reporting the methods and results of an observational study, right? So you can literally just go down the checklist and make sure you're hitting all the components that the Strobe guidelines have recommended that we include. And in fact, many of our journals, many of our top journals, actually will require a completion of this checklist when submitting your manuscript for publication. So I think it's, in and of itself, it is a great set of guidelines and should always be used. But again, from a practical perspective, sometimes it's good to get out ahead of this and go ahead and complete the checklist as you write your methods section. And here is a look at the version 4 of the Strobe checklist for a cohort study, just to give you a feel for some of the information that is actually on the checklist and what this looks like. So as you can see here, these are a list of points. I've omitted the first 3, but you can see here when you come to the methods section, these topics mirror very closely to the slides that I've already shown you about some of the general sections, or subheadings of the methods section, right? There's not an exact one-to-one correspondence, but generally these are conveying the same areas that you want to include in your methods section. So for the study design, they recommend that we present key elements of the study design early in the paper. I agree, often this is my first subheading in the methods, and the first sentence there is a description of the study design. Here in the setting, as we touched on before, we want to describe the setting, the location, the relevant dates, the periods of recruitment, exposure, follow-up, and data collection, right? We want to give sufficient detail around all these aspects for a reader to understand exactly what was the setting in which we carried out our research study. For participants, we want to be sure to give the eligibility criteria and the sources and methods of selection of participants. And then here for match studies, certainly give the matching criteria and the ratio of exposed to unexposed. Or for cases and controls, it could be a ratio of cases to controls, for instance. For the variables, as I said before, we wanted to clearly define all of our outcomes, our exposures, predictors, potential confounders, and effect modifiers, right? So anything that's going to come into our statistical analysis, we want to be sure that we describe it, how we obtained that data and how we used it in our study. For data sources, here they recommend for each variable of interest, give the sources of data and the details of methods of assessment, right? So that's exactly what I recommended before in terms of that section. And then to describe the comparability of assessment methods if there is more than one group. In terms of bias, again, describe any efforts to address potential sources of bias, and for the study size, to explain how the study size was arrived at. And so often this is, again, in the form of something like a sample size determination, which I'll touch on in the next webinar. So I am certainly not the first person to think about and to provide a video on effective ways to approach the writing of the method section of your academic or clinical research manuscript. So here I just wanted to include a few additional resources that might be of interest to some. Here under the recordings are 2 different links to other YouTube videos that might be helpful or informative in terms of helping you to think about how you might want to structure or approach the writing of your methods section. And then here under the papers, I'm giving you a link to a paper that actually touches on a little bit about how we might want to write a method section, what you might want to include. And then, as I said on the last slide, a link to the Strobe statement. This link will take you there and you can access the Strobe recommendations as well as obtain the Strobe checklist that I showed before. And so now we come to the next step for our ARC investigators. And for you, it is now the time and the opportunity to begin to think about and to start drafting the methods section of your manuscript. And when you do have that methods section developed, or at least a first draft or iteration, those materials can be submitted via email to the ARC Diagnostic Network email. And similarly, if any questions do arise, be sure to reach out to us at the ARC Diagnostic Network email with any questions that may come up. Again, don't hesitate to contact us if you need any assistance or have any questions with any aspect of drafting the methods section of your paper. We are here and we're happy to help. And also, please inform us of any comments that may help us to improve future webinars. And with that, I look forward to seeing you all again in our fourth and final upcoming webinar. And thank you all again for your continued work and contributions to the ARC Network.